There we go. Good morning. We have several class members traveling today for different things, and so I'm going to be recording this lecture. And before we get started, I have graded lab exams. We're going to go over them tomorrow because I've also saved the materials from last week in case you want to look at this tube, that tube, and so on. So tomorrow in lab, we will meet at the normal time in the classroom and start out by describing how you work on your unknowns. And then we'll go down to the lab and I'll give back lab exams and you can look over those and also start on the unknowns tomorrow. So I think there are some materials in Canvas that describe how you do unknown reports, and those are 95% true for this year. There's just a few things that will be a little bit different if you want to look over those ahead of time. I think that's all the announcements for today. This is a more relaxed week in microbiology, so minimize that. And today our topic is phototrophy, photosynthesis in microbes, specifically in prokaryotes. And we're going to be looking at two major styles, which are anoxygenic, without making oxygen, and oxygenic. But both of the styles of being a phototroph, the energy source is light. So light, and it's going to be specifically sunlight, is going to be the energy source. And all phototrophs have that in common, but what they use as their carbon source can be different. Most of them are going to use CO2 as their carbon source. And this would include plants, algae, cyanobacteria, most of the major groups except for one. And there is this one group that we are going to look at later today when we describe the major types, called heliobacteria. And heliobacteria are different in that they can use sun for energy, but be using organic carbon for their carbon source. So it's kind of like they are using sun for energy and something that we would think of as food for their carbon source. And they would be an example of what we call photoheterotroph, or when you see later in the slides, there will be some type of description that this particular type of microbe can do what we call heterotrophic growth. That's what that means, that they can use organic carbon for their carbon source while they are also using sun as the energy source. But most photosynthetic life on Earth is doing this style. This style well, let me go forward from this slide here. Okay, so that's how we could classify phototrophy according to the carbon source. We can also classify phototrophs as to what style of photosynthesis are they doing. So the first style and the oldest style is called anoxygenic. And it's named this because of what the waste product is. So in this style of photosynthesis, the major waste product tends to be a sulfur compound. Usually some version of a sulfate. And because this is not oxygen, this style of photosynthesis is called anoxygenic. The types of microbes that do this are purple, they're called purple and green bacteria, and we'll look at more details about how those operate later. But they're making a waste product which tends to be sulfate. They are using CO2 as their carbon source. They are doing all of this powered by light to make ATP. What's different about these is the waste product and also these two things. So I'm going to put these in a different color. What this chart is calling reducing power. So organisms that do anoxygenic photosynthesis, they would need these in large amounts. Because this is where they're getting the electrons to keep replacing electrons that are being excited by light. And then eventually these compounds, once the electrons have been removed from this, it becomes the waste product. So we can call this reducing power, or we can call this the source of replacement electrons. So organisms that do anoxygenic photosynthesis need large amounts of hydrogen sulfide, or sometimes large amounts of just plain old elemental sulfur. They would need that in large amounts. These would have existed on early Earth and been around in great supply, and that helped this style of photosynthesis develop first, the anoxygenic style. Because of the tendency to need hydrogen sulfide or just sulfur in large amounts, these tend to not just be called purple and green bacteria, but purple and green sulfur bacteria, because they rely on sulfur so much for their metabolism. And this is anoxygenic style. One more thing we'll put on here. During this photosynthesis, we could say that these organisms are fixing they're doing carbon fixation, or they're fixing CO2 and building it into organic compounds. So that's similar to how a plant works. Pull CO2 out of the air, use sunlight energy to make ATP that then allows them to build large organic structures. That part is still working the same in an anoxygenic photosynthesis. So let's kind of compare that to oxygenic photosynthesis. And I'm going to kind of erase some of this slowly because I want this to match up with how we describe the anoxygenic ones. Okay, so let's start here. Oxygenic photosynthesizers are also using light energy to help them fix CO2. Basically, pull CO2 out of the air, use the ATP that they get from absorbing light, and build big organic compounds. Still true. What's different is the way that this is named. It's named oxygenic because there is a major waste product produced here that is oxygen gas. So the major byproduct of this style of photosynthesis is oxygen gas. And it's weird for us to think about this as being a waste product because it's so essential for our life. But it originally was truly a waste product. And this waste product, we spent a lot of time talking about last week, like how this changed the world. So as this built up in the atmosphere, that led to ozone. It led to banded iron formations. It led to the option for aerobic life. I know that's a lot of low down writing, but when we talked about the great oxidation event or the appearance of oxygen on Earth, it came from oxygenic photosynthesis. And so long ago, the buildup of this waste product, it made all of this other stuff possible. So for this style of photosynthesis, what is needed in large amounts is water. So we could say water is the reducing power. Water is that source of replacement electrons. Or we could say that you need constant supply of water to keep this going. And this is the style of photosynthesis that you started learning about, when you were younger. As this is how photosynthesis works. Pretty much still true in microbes, but the only microbial group, the only prokaryotic group that does this style is cyanobacteria. Of all the prokaryotic groups that do photosynthesis, most of them don't do this style. This is the advanced, newfangled in the history of the Earth, exclusive, special photosynthesis that produces oxygen. Plants do it, algae do it, but in the prokaryotes, it's just cyanobacteria that do this. And they're still doing this. There are literally tons of cyanobacteria on Earth still doing this. But most prokaryotes that do photosynthesis do anoxygenic. Cyanobacteria do the oxygenic style. So these two slides are a really good comparison. Like, what are the big differences between these styles? The oxygenic style is what you learned about before in previous classes. So knowing that, I want to kind of back up now and review, like, what are the major things besides this stuff that you probably learned about before with plant photosynthesis, and how do plants do this? Then we can compare what's different with how microbes do photosynthesis. So one of the big key concepts that I will want you to know for the exam on this section is what major ways does prokaryotic photosynthesis differ from eukaryotic photosynthesis? And we have a list in this slide set that's a little bit spaced out. I think it has 5 or 6 things that are different between them. And that's like a listing type question that I want you to remember for the lecture exam on this section. So what are the big differences between how prokaryotes do photosynthesis and how eukaryotes do photosynthesis? So that's where we're heading next. Okay, so let's kind of look back. How do plants do photosynthesis? And algae as well as eukaryotes. So one thing that you tend to learn when you learn plant photosynthesis is the concept of a chloroplast. Learning that's where photosynthesis happens. You've got this separate organelle. And this chloroplast organelle has multiple membranes associated with it. So you've got inner chloroplast membrane, outer chloroplast membrane, and a thylakoid membrane. And that's where photosynthesis tends to happen. And the innermost membrane tends to be kind of stacked in these grana configurations. So we could say that the most internal membrane is very stacked or very folded, which increases that surface area. So the more folding, the more stacking inside the chloroplast of these membranes, the more light harvesting complexes you can get in there, and the more efficient this becomes at gathering a lot of light in a small amount of space. So that's one characteristic of how eukaryotes do photosynthesis. And the other thing to remember about chloroplasts is where they originally came from. Where did chloroplasts originally come from? I think only Liz Midbed in this class. Or did you have plant bio? Well, you don't have to have plant bio to answer. Yeah, what were you gonna say?
Like something that was ingested at some point?
They were engulfed
Yeah.
at some point.
And they became.
And they eventually, over time, became a chloroplast.
So is it the endosymbiotic theory?
Yeah. This is endosymbiotic theory, which I kind of skipped over in this unit. So remember, chloroplasts, this is, like, it's said that this is a theory. It's not a theory like we think this may have happened. Really sure that this happened. So chloroplasts in a modern sense, came from a photosynthetic prokaryote engulfed by a eukaryotic cell, thus giving you an organelle that has its own genome, its own set of membranes. It's the size and shape of a phototrophic prokaryote. And the chloroplasts themselves have a lot in common with photosynthesizing prokaryotes. A lot in common, which we'll see examples of that as we go. But having a chloroplast is a big characteristic of how eukaryotes do photosynthesis. The other major characteristics that we need to review are going to be on this slide here. So just taking a look at this for a moment. This is depicting a chloroplast. What we've got in the cartoon here on the screen is a chloroplast. This would be the stacked membranes of the phylloploid. And those are absorbing light as the energy source. The energy from the light is used to eventually store energy as ATP, store energy as NADPH, then fix carbon. So we would call this part carbon fixation into building organic compounds. Light is the power source, CO2 is the carbon source. The water is the source of replacement electrons. The water is needed in great supply to keep this going. And then we have oxygen produced as the waste product. So this has the same info that we saw on the flow chart, just with the picture, which is nice. Something else that is different about this is that in chloroplasts, we kind of have two divisions of things going on that we call the light reactions and the dark reactions. So the light reactions are making stored energy. The dark reactions are doing the carbon fixation. But all this is happening in a chloroplast. And this is oxygenic style. So as far as we know, all chloroplast-based photosynthesis is producing oxygen. It's oxygenic style. So what we're going to go through next is, okay, what are the 5 or 6 big differences from this that prokaryotes use when they are doing photosynthesis? So what are the big differences with how prokaryotes do photosynthesis? The first one is that prokaryotes don't have traditional organelles and they don't have chloroplasts. So they can't use chloroplasts to do photosynthesis. And that's kind of basic and maybe obvious, but what this means is if you don't have a chloroplast to hold your photosynthetic membranes, you have to have all of your photosynthetic equipment in the plasma membrane. So all of your light harvesting proteins and your electron transport processes, all of your photosynthetic equipment has to be in the only membrane that you've got, which is your plasma membrane. Or let's call it the cell membrane. And this picture is from a different book than the one that we use, but want you to know what membrane this is. So we've got cytoplasm on this side. This side is more toward the outside of the cell. This would be technically a gram-negative because it's got paraplasm out here. But all of the light harvesting equipment, the ATP synthase that you eventually need, all of that is in the regular cell membrane, not in a chloroplast. So that's probably one of the biggest major differences. There are no chloroplasts. Everything you need is just in that one membrane. Details of what is in the membrane are less important for this class, but this is a very important fact. Next. Oh, before we move on from this, one thing that is the same is that there is still, this equipment is still forming a proton gradient. That's not different, that's the same, just the proton gradient is forming outside the cell. And it's still going to run through ATP synthase, it's still going to make ATP chemiosmotically. I'll write that up higher. We're still having chemiosmotic ATP production. We're still using a basic ATP synthase to do this. Okay, next thing that's different. And this is going to be like, what? If you haven't had plant biology class this year. But one thing that is different about the way that electrons get excited and their energy used in prokaryotic photosynthesis is that it uses something called cyclic electron flow, which is shown here. So I'm going to try to explain this in a clear manner. So there is a pigment, protein complex that is able to absorb light in these microbes. And the particular one shown in this figure is called P870. The name of it is not too important for this concept, but that's what P870 means. And it would be equivalent in concept to something like this. So there is a protein associated with the pigment and it can absorb light. And the light that gets absorbed into that complex excites an electron. So we see this is the redox potential of that complex before light hits it. Light hits it, and we see this arrow going up. And then we see this now being called P870 asterisk. What this means is that this has an excited electron. Basically, light hit the pigment, it made the electrons start moving faster, and now we have an extra energetic electron that has been raised up to that level, and we put an asterisk to show that. That electron now has more energy than it did because it got boosted by light. So now this becomes a good electron donor where it wasn't before light hit it. And this excited electron then is going to move through a type of electron transport chain. And it's shown here, okay, it moves on to this chemical, which is bacteriochlorophyll. Then it passes to this one, which I'm not sure what that name is, through some quinones. through to a cytochrome, and eventually that electron will make it back to the same place it came from, back onto this pigment protein complex, P870. This is called cyclic electron flow. So in plant biology, reach back, what is the shape that we tend to draw electron flow for a plant? It's called a blank scheme. It's not a circle, it's something else. So few seniors in here. That's too bad, because this stuff is a good review for your MPAT exam, like your biology comprehensive exam. So in plants, we have a Z scheme. You ever heard of that?
No.
Z scheme? That's a Z that I tried to write there. Z scheme of, well, there's like a double boost of electrons. There's first absorbed some amount of light, raise up to this level, absorb some more light, raise up to this level. And the electron at the end gets passed on to some different molecule. It doesn't return to the beginning. So cyclic electron flow means the electron, you can think of it as it eventually makes its way back to the same spot, but the energy boost that it got from light is kind of siphoned off by it passing through an electron transport chain, and its energy gets turned into a proton motive force. So what that is, that may sound like, I don't know what that is. You do know what that is. That's a proton gradient. Proton gradient. So light hits a photopigment complex. The electron gets energized. It gets passed down the chain from one place to another. It goes through a cyclic flow. That energy eventually gets turned into proton gradient, and that runs through ATP synthase and gives ATP. That cyclic flow is different from what plants do. Plants do a Z scheme. Okay. Oh, here's another picture of it showing light excitement, electron flow. It moves in a circle. You eventually make ATP. For this class, the details of what the proteins are and what they're called and what these players are not a big focus for this class. Just cyclic electron flow as opposed to Z scheme electron flow. So that's a big difference. Another big difference with prokaryotic photosynthesizers is that they tend to, what I call here, contort their membranes. This is not a moment-to-moment decision. Like, I think I'm going to contort my membrane today so I can harvest more light. More like this. Over time, they have evolved to have membranes with greater surface area. Membranes with greater surface area in a variety of ways. So what you're looking at here is each one of these is a cell, a prokaryotic cell that can do photosynthesis. So this is not a chloroplast, this is a cell. All of these are prokaryotic cells. And prokaryotes that do photosynthesis tend to have one of these styles of membrane surface area expansion. So some of them will have tubes that kind of extend toward the interior. Some of them keep their membranes in a shape that's kind of like interior budding vesicles, like this shape. Some of them have bundled tubes, which are like multiple of these, but stretching into the interior. Some of them have stacks, like so. This one looks like granite. That looks like thylakoid stack, this configuration. And then some of them do like this lamellae looking thing. The benefit of this is that the more extended your membrane, the more of all of this, I like the other picture better. The more of all of this you can fit into the membrane. You can fit more light harvesting complexes. So if you think of photosynthetic equipment like this, that if you're a bacterium, if you're a prokaryote, and the only place you've got to put your photosynthetic equipment is in the membrane, the more membrane you have, the more light you can gather. So it's like if you're gonna have a solar farm, you need to spread out your solar panels over as much surface area as you can, because the more surface area, the more ATP you're gonna make. So membrane contortions or folding or anything like this that increases the surface area will be beneficial. And just to kind of make clear what this is meaning, this is one membrane and it's just folded like this over and over again. Just with huge folds and surface area increases. And that's really beneficial for microbes that are going to photosynthesize, get more surface area. And then right before our break, we're gonna have our break a little bit early today. We've got some pictures of prokaryotes that do photosynthesize, and this is showing what their membranes are like. So this one, it looks like it's full of little bubbles. It's actually all one continuous membrane. This is equivalent to this when you look under a microscope. And here we've got an example of lamellar membranes. So here we see the bacterium, this is its plasma membrane, and the plasma membrane is continuously folded on the interior into these lamellae structures. And these kind of look like chloroplast folded membranes. Okay, so let's have our break right now. And then we'll return to what are these big differences? And we'll kind of also get into some, kind of depart from that list a little bit to describe some cool things that the photosynthesizers can do. Okay. Okay, so we have about halfway through, how are prokaryotic photosynthesizers different from plant and algae photosynthesizers? And one big difference in how they do photosynthesis is what they are using to absorb light. And we have up here a picture of one version of plant chlorophyll, one version of bacteriochlorophylls. So both of these, I do not remember which one is which one. Both of these are chlorophylls. Both of these are based on a heme-based compound, which is kind of like hemoglobin. So we have a set of ring compounds, and they have a coordinated double bond system. So this can absorb light, this can absorb light. If this was hemoglobin, what would we have here instead of magnesium? Iron. Yeah. So these heme type structures are really important for many types of life. If there is a magnesium at the center, that helps this be a chlorophyll. So magnesium is essential for building these. So what you're seeing in the yellow, the light yellow, is the same between these, but there are some different R groups. Between these two types of pigments. So what they've got shaded in yellow and blue are where the differences are in these two versions of it. So you need a pigment to absorb light. The pigments are going to be different from each other based on what are the R groups, and having slightly different versions give you the ability to absorb light of different wavelengths. Absorb. We need to absorb light of different wavelengths. And this makes sense for prokaryotic photosynthesizers, because prokaryotic photosynthesizers are almost always in water. So if you think about, okay, how did photosynthesis ever develop? It was back in the days before there was oxygen. The land would have been deadly to life because there's no ozone to block the UV rays. This would have developed in water. So if you are photosynthesizing in water, depending on how deep you are, the wavelengths of light are going to vary according to the depth of the water column. So you would need different types of pigments that help you absorb different wavelengths of light. So they're going to use different photopigments. These types are types of chlorophylls. There are other pigments that are not chlorophylls that are also important for prokaryotes and eukaryotes to absorb light. So we'll look at those next. Okay, so what we've got here is something called absorption spectrum, which is a graph. On the x-axis, we've got what is the wavelength of light. So this is representing the light wavelength in nanometers. And on the y-axis, we've got absorbance, which is asking how well is the light absorbed? And where you're seeing, the numbers are a little bit messed up on here, but where you're seeing high value up on the y-axis, that means that that pigment is absorbing that light well. And if you see a trough or a dip in one of these lines, it means that that wavelength is not being absorbed very well. So this is showing the difference between regular chlorophyll A, that's a version that plants use, and a version called bacteriochlorophyll. So we've got chlorophyll A versus bacteriochlorophyll A. And these two pigments are these two pigments. It's showing that having those different R groups make these absorb a little bit differently certain wavelengths of light. So if we look at regular chlorophyll A, it's absorbing well around 450. It's absorbing well at 680 nanometers. Not so well in between, not so well at the short wavelengths, not so well at the high wavelengths. Bacteriochlorophyll is absorbing well at the short wavelength, and it's absorbing well at the long wavelength. So what this means is that these two pigments are specialized to absorb different wavelengths of light or different colors of light. So you can write underneath this, we can think of this as colors. Colors of light, because this is close to the visible spectrum. So think about what you learned about visible spectrum of light, what the colors are. Did you learn Roy G. Biv as the acronym? I did. Red, orange, yellow, blue, sorry, green in there, G. Blue, indigo, violet. Is this end closer to blue or red? This is closer to the blue end. So the shorter wavelength light is closer to blue/UV. This end up here is closer to red or infrared. So red wavelength of light, red color of light has a longer wavelength. Blue color of light has a shorter wavelength. And that is related to this water column situation. So we're going to come back to that in a couple minutes. But depending on what light is in your environment, you better have pigments that can absorb that light if you're going to be successful at photosynthesizing. And you can measure this stuff on this type of graph called absorption spectrum. Okay, so there we have chlorophyll-based pigments. We also have pigments that are more of a linear conjugated double bonds. So we've got here conjugated double bonds. We've got here also conjugated double bonds, and that tends to characterize light absorbing pigments. So when you have conjugated double bond systems, essentially those are going to allow light to get electrons moving. So when light hits a conjugated double bond system that has particular R groups associated with it, light of different wavelengths can get electrons moving, can get electrons excited. So having conjugated double bonds helps with light absorption for that reason. This version of a pigment, which can absorb light, get electrons moving, is called a carotenoid. So carotenoids are pigments that tend to have a linear structure, and carotenoids plus chlorophylls, if you have both of them together, those are going to maximize light absorption. So most organisms that do photosynthesis, they don't just have one pigment, they have a set of pigments that will help them absorb the wavelengths of light that they are likely to be in. So organisms would not have just carotenoids or just chlorophylls, they would have sets of them that together absorb the light that they're in. Carotenoids also provide photoprotection. So by photoprotection, they are going to absorb the possibly harmful wavelengths, which tend to be up in the blue or the UV spectrum. Like that's high energy light. That can be more damaging. Even if we've got ozone layer to help block those, it's still good to have photoprotective pigments to absorb that slightly dangerous, slightly too energetic level of light. And carotenoids can do that as well. And for us, one of our major carotenoids is beta carotene that we eat in yellow, orange fruits and vegetables, and it helps provide our eye pigments and also some photoprotection for us. So photosynthetic microbes can have chlorophylls, they can have carotenoids, and then they also have something called accessory pigments. Spelled right, accessory. Accessory pigments to also absorb light. And I want to, on this, there's a lot going on this slide, so I want to start here. This is the membrane of a photosynthesizing bacterium. It would be a cyanobacterium. So we could add that word in here. This is a membrane. Would not so much call it a thylakoid membrane, it is a microbial membrane. And it has in the membrane some photosystems, and out here, it has created something that we will call the antenna complex. So this is a set of proteins and pigments that the bacterium has built in conjunction with its main photosystems in the membrane. So these are harvesting light, but this is an assistant or an antenna complex to help harvest more light. So this is set up to where it's going to absorb light, absorb wavelengths, and it acts to funnel that light energy down to the main photosystem. If you've ever looked at all these things, like a satellite that goes into outer space and then it It shoots out there and it starts drifting through space, and then it has this large foldable solar panels that come out and they unfold, and it's to help the little craft gather light to power its flight through space. That's kind of like this antenna complex. So this is an assistance to help gather more light. And in this complex, there are accessory pigments called phycocyanin, or it can be called phycocyanobilin. There can be blue versions of pigments out here in the antenna complex, or there can be the red version. So these two are chemicals that can contribute to this antenna complex to help assist with gathering light. And we want to add on here their abbreviations. So this version is called phycocyanin, or PC, and it is a blue pigment. So they're showing a blue color of this box here. This version is called phycoerythrin, or phycoerythrobilin, PE, and it's a red pigment. So in these boxes, they're showing what's the chemical group on this one. And they've also made them a color, and the color matches what the pigment looks like. So make a note that phycocyanobilin pigment that you could have in this antenna complex, it appears blue. Phycoerythrin appears, if you were to look at this with your eyes, it appears red. Okay, so this section, talking about how do prokaryotes decide what pigments and how does that relate to where they are in the water column. This is a little bit of a problem solving or a physiological concept, and it relates to this water column idea. So if you are a microbe and you live in water, you may or may not be able to make yourself swim up and down to follow your favorite light wavelength. And the light is going to be coming from the sun, as it always does in nature. And sunlight, as it hits the water, it might be nice to have colors here. There are different wavelengths and different colors of light that are associated with visible sunlight. So one of them is blue light. And blue light is short wavelength, and because it's short wavelength, it does not penetrate very low in the water column. So blue light is going to be kind of most at the top, the shallow, underneath the surface of the water. Red light, this one needs a purple marker to represent red light. So this is the blue end of the spectrum. The red end of the spectrum, that'll work, it's reddish. Red light, because it's longer wavelength, penetrates further into the water column. So if you're a microbe that photosynthesizes and you can adjust your pigments to deal with the light that you find yourself in, that's really a good advantage. Microbes that can make both of these pigments have an advantage over ones that would not make both, because they could adjust their light harvesting to deal with what light they're in. So there are certain microbes that can do what is called chromatic adaptation. Chromatic adaptation. So if you're a microbe and you can do chromatic adaptation, what that means is you can change your pigments to harvest multiple wavelengths of light. Not all photosynthesizing microbes can do this, but the ones that can have an advantage. So if you are such a microbe and you find yourself down here, so there you are, you're down there, what light is available to you? You are in a red light region because you're too deep to get the blue light. You're in a red light region. So if you're down here, you need to absorb red. That makes sense. If you are up here, you are in a red and a blue light region. Which one would be better to absorb? It is blue. Why would it be blue?
Higher energy.
Blue light, if you can absorb it, if it's there, it's a little bit more energetic than the red light. So it would give you a little more bang for your buck for photosynthesizing. So if you're up here, you would prefer to absorb blue. But you may not necessarily be able to swim yourself up or down. It's kind of like, okay, what light do I have right now? Okay, it's blue, so I need to make myself a blue absorber versus I need to make myself a red absorber. So we've almost got like all the info that we need to understand this. The final thing is putting in which pigment do we need here, which pigment do we need here. And before we do this, box or circle or underline the word absorb. So it's not that these bacteria need to look red, they need to absorb red. Now, think back to what you know about plants and why they look green. Why does a plant look green? Because it absorbs everything but green and it reflects green. So the way that you look is the opposite of what you actually absorb pigment-wise. So let's go back and add in here, okay, if you are a pigment that appears to be blue, what you actually absorb is red. And because you absorb red, you don't reflect it, you reflect blue, so you look blue. So phycocyanin type pigment looks blue, and that's where the name comes from, phycocyanin, like cyan blue, but it absorbs red. And the opposite is true for phycoerythrin. It appears red, but it absorbs in the blue spectrum. So knowing that, now go back onto your water depth picture here and think about, okay, if you are in blue light, what pigment do you want to be making?
Red.
You need to make a red pigment, which is called PE, which is phycoerythrin. So erythrin, that's like erythrocyte, like red blood cell. So you need to make phycoerythrin if you are finding yourself in blue light, which is shallow water. But if you are in red light and you need to absorb the light that you got, which is red, you should make a pigment that absorbs red, but which will appear blue. So you would want to make phycocyanin pigment. And the ability to do that is called chromatic adaptation. It's a pigment switching. Pigment switching. And what they're switching, they're not creating a whole new photosynthetic membrane. They are switching what do they put in the antenna complex. Is it more phycocyanin? Is it more phycoerythrin? So on the test, there are like this type of situation, it's kind of like a 50-50 thing, any question I ask you. So examples of questions that I might ask are, like they're always going to have one of two answers. So example, if an organism is in deep water, what I call deep water, which light is most abundant down there? Which color of light? Red. Another example of a question is if the bacterium is in shallow water, what pigment should it be making more of? Have to think about this. So if it's in shallow water, there's blue light up there, it would like to absorb that, it ought to be making a red pigment, which is P. The third type of question is, what color would one of these cyanobacterium appear to be if it's in deep water and it's doing chromatic adaptation? What color would it look? Bluish greenish. Yeah. So I will be asking questions like that on the exam. They are not like it's a 50-50 shot. It's blue or it's red. It's phycoerythrin or it's phycocyanin. It's deep or it's shallow, things like that. But it's kind of a concept. And just when you're working through this, like to me, I tend to get stuff backward if I'm not careful. So having this kind of picture is helpful. Like, which light goes deep, which light stays shallow, what color are they? And remember that whatever color you look, it's because you're absorbing the opposite. So, you know, you can make up questions like this and quiz yourself. Like the type of questions are, what color is the microbe? What color is the pigment? What color of light does it find itself in? And which, you know, what would it prefer? You always want to absorb the light that you have available. If you've got a choice, you would want to absorb the light that's more energetic, which is the blue. Chromatic adaptation. It would be nice if we, well, sort of say, it'd be nice if we could get plants to do this, but plants are not so much under the water. This is a pretty hot research topic because if we are going to light harvest in space, it would be from microbes that are growing in water tanks and getting different wavelengths of light in space. So, research topic. Okay. So I forgot I had this cartoon. This is just showing what it would look like for microbes to do chromatic adaptation. So if you find yourself in red light, you need to make yourself look blue so that you can absorb red. This is just showing it with color. Can you tell these are different colors? Yeah. So just, if you have slides, make yourself a note. This is called chromatic adaptation. The ability to do this. And it takes them maybe some number of hours to do this switch. So they're sensing what is the light color. And then they change their gene expression to make more of one or the other. Chromatic adaptation. Okay, this slide we're going to skip over. It's true facts, but we're gonna skip over that. Okay, so we're now returning to, okay, what are the differences between plant and prokaryotic photosynthesis? And one of the major differences is that where the replacement electrons are coming from can be different. So it says here they are different, they can be different. That doesn't always have to be water. There can be other options of what is this photosynthesizer using in large amount to do replacement, to give replacement electrons. So this chart is actually describing not where the replacement electrons are coming from, but what are the pigments? What are the pigments in the different bacterial groups? It's the next slide that shows what are the replacement donors and the waste products. So make a note. This chart is talking about what are the pigments. What version of chlorophyll? The next table is showing what are the replacement donors and what are the waste products. Okay. So I'll start here. This is showing in this first row, what is the source of reducing power? That's from the previous slide. Number 5, where do the replacement electrons come from? It can be water. If we're talking about cyanobacteria, we would put plants in that section also. But it can be other things as well. So it can be hydrogen sulfide gas. It can be regular hydrogen. It can be sometimes an organic thing. It doesn't have to be just water. And on number 6, this number 6 difference, sometimes prokaryotes are doing anoxygenic photosynthesis, which means that the waste product is not always just oxygen. So this aspect is matching with what this chart is calling. What is the oxidized product? What is the waste product? If we're talking about plants and cyanobacteria, the waste product is oxygen. Because water was the electron donor, and when electrons are removed, the waste product is oxygen. If the electron donor is something that's not water, the waste product is something that's not oxygen. And a lot of times it's going to be sulfate. That's a big difference between plant-style photosynthesis and microbial style photosynthesis. So what is the source of replacement electrons? That's this row. What is the major waste product? That's this row. And before we kind of leave this concept, make a note about cyanobacteria. So cyanobacteria do photosynthesis. Their photosynthesis is most similar to plant photosynthesis. Cyanobacterial photosynthesis has a lot in common with plants. So both of them are using water to replace electrons. Both of them are creating oxygen as a waste product. They are using fairly similar pigments, like similar styles of pigments, but they don't have chloroplasts. They don't have chloroplasts. They don't have chloroplasts, but they are definitely the ancestors of plant chloroplasts. We're going to stop there for today. There is still some more info in this slide set about, these are the 5 major groups of prokaryotic photosynthesizers. We will do that info on Thursday, as well as you'll want to bring new slide set for fermentation and anaerobic respiration, which will be a short one, which will appear today or tomorrow on Canvas. So don't forget, tomorrow we'll meet for lab at 2 p.m. in the classroom. Talk about working on your unknowns project and how that will go, and also give back lab exams tomorrow.
Is our test a week from Thursday?
I think so. Seems like it's, well, I gave my other class 2 tests last week, so it feels like to me like tests everywhere. But yeah, for this class, lecture exam would be next Thursday.